Welcome to Sir Robert Koch Tutorials. Learning is 24/7. Hello and welcome to Sir Robert Koch Tutorials. Today I want to look at the history of microbiology and parasitology. History is very important. You know that in your study of history, the definition of history was the study of past events or what happened in the past. And some of the reason why we study history is for us to know our enemies and our friends, and in this case our enemies are the diseases and of course the microorganisms that cause infection. These are our enemies. And the other reason why we study history is because we want to know our past, our present, and our past and our present in order to improve our future lives. So we need to know the past of this of microbiology and the present of microbiology and how these microorganisms are evolving and how they are affecting us, so that we are able to plan for tomorrow to improve our health lives. So during the biblical times, it was known that some diseases are infectious, right? For example, leprosy is one of the diseases that characterize the greater part of the Old Testament. You will see that in Deuteronomy, in Exodus, in Leviticus, and all other verses that are in the Old Testament. It is documented vividly, and you can see that leprosy was one of the diseases that was being mentioned, and mostly it was associated with sin. When someone sins, then they are affected by leprosy. So it was believed that if someone sinned against God, leprosy was a form of punishment which was later to visit that person. So disease during that time was a visitation of God's wrath against iniquity and people who sin against God. So centuries later, the diseases were then identified now as the causative organisms that are associated with causation of diseases, not sin. So syphilis, leprosy, and plague were identified as the main causes of disease in the Middle Age. Ancient humans ascribed it to supernatural forces, like I indicated earlier on. Those who usually caught lepers were characterized as people who were very sinful in their nature. Some pioneers in microbiology. Let us look at pioneers of microbiology, people who studied the work of microbiology. We had a great historian, we had a great microbiologist, Anton van Leeuwenhoek. Anton van Leeuwenhoek. Anton van Leeuwenhoek lived from 1632 to 1723. Anton van Leeuwenhoek from 1632 to 1723. He built a microscope in 1675. Microscope was just a lens held on a metal clip. You can see how this work started. His microscopes would magnify about 50 to 300 times. That was during his time. He observed animalculae in water, soil, and human. So during this time, the microorganisms were being named as animalcules. They were being called animalcules. So he called them animalcules, right? So animalculae in water, soil, and humans. He was the first person to identify bacteria using a microscope, Anton van Leeuwenhoek, right? Robert Koch. We have Sir Robert Koch. Sir Robert Koch is yet another great person in the history and development of the field and branch of microbiology. He was a German practitioner. He was from Germany. He discovered and cultured the bacteria that cause disease, including tuberculosis. So he is the man behind the discovery of this bacterium called Mycobacterium tuberculosis. So Sir Robert Koch played a critical role in the identification of Mycobacterium tuberculosis. So this is what we learn in this issue of culturing. He used the Leeuwenhoek's microscope to see bacteria and draw the structure of some microbes. So this is what he did. So he built on the work of Anton van Leeuwenhoek's microscope in order to see bacteria, to visualize it, and also to be able to identify and clearly draw the structure of the microscope of the microbes. So basically, he laid down the criteria for confirming an organism as the cause of disease. So he's the first person to actually lay down the foundation upon which today we are confirming that this is TB, this is gonorrhea, this is syphilis. It was because of the work that he started. He enabled us to have a criteria of confirming an organism is the cause of a disease, because he started the process of culturing in order to identify and really understand any microorganisms that is behind a particular disease. Another most important person is Edward Jenner, Dr. Edward Jenner. In 1796, he introduced vaccination. Now we've got vaccination being introduced by Edward Jenner, Dr. Jenner. That was in 1796 when he introduced vaccination. He also showed that smallpox was preventable by inoculation with the related disease, cowpox. So Dr. Jenner is the one who discovered that smallpox could be prevented if someone was to be inoculated with the related disease, cowpox. Why? How does that work? Because the related disease, cowpox, right, the attenuated virus in that regard, it sensitizes the body to fight against the future encounters of the same infection. Generally, that's how the immune system works. It works by sensitizing the body. The body is able to recognize an encounter of an antigen when it starts invading the body. The first vaccine of smallpox used the smallpox vesicles. How he did not discover the cause of action of the infection. Now I want to look at Dr. Louis Pasteur. Louis Pasteur is the one behind what we call pasteurization. Louis Pasteur is the founder of modern microbiology. He existed from 1867 to 1888. He developed methods of culturing bacteria. He also established the principle of the preparation of vaccines against the infectious disease. Then we say Alexandra Flemming. He developed penicillins in 1929 from a fungus penicillins. So this is the man behind the penicillins. For us to say this is amoxicillin, this is benzoyl-penicillin, this is cloxacillin, the penicillins were developed by this man, say Alexandra Flemming. That was in 1929. He developed it from a fungus penicillins. However, Flemming had little to do with developing penicillin as a treatment for infectious disease. After some initial experiments, he abandoned the study of its activity. Fortunately, Ernst Chain developed an interest in the chemical and eventually purified it. In 1940, in World War II, Howard Florey and his assistants began successful clinical trials with penicillin. By that time, Selman Waxman had discovered several other antibacterial agents and had coined the term antibiotic. After success of penicillins, many antibiotics were developed. So the first antibiotics to be developed were the penicillins. And then we've got Joseph Lister. Joseph Lister was a professor in surgery. He followed up Pasteur's work of fermentation. He tested if fermentation of sugar solution in starch was due to microbial action. He also attempted the prevention of microbes in wounds and injuries, including main forms of sepsis. He used phenol to destroy germs. He also demonstrated that wound sepsis was preventable through antiseptic techniques with carbolic acid. So today, that's why we've got antiseptics that we used to treat against the septic wounds. He is the first person to design techniques of obtaining pure cultures. Now we want to look at different people who are behind the discovery of different diseases and their causative organism. The people who are behind the discovery of certain diseases and their causative organisms, and of course including the years in which those organisms were discovered. So we're going to look at the year when the disease was discovered, the disease that was discovered, the causative agent of that disease, and the discoverer, who is the person who discovered that particular condition. So in 1876, anthrax was discovered by Koch and the causative agent is Bacillus anthracis. In 1879, gonorrhea was discovered by NASA and its causative agent is Neisseria gonorrhoeae. In 1880, typhoid fever was discovered by Gafke and its causative agent is Salmonella typhi. In 1880, malaria was discovered and its causative agent is Plasmodium species. You know, with Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and the discoverer was Leveran. In 1882, tuberculosis was discovered and its causative agent is Mycobacterium tuberculosis and the discoverer was Sir Robert Koch. In 1883, cholera was discovered and the causative agent is Vibrio cholerae and the discoverer is Sir Robert Koch. In 1883 to 1884, diphtheria was discovered. Hello and welcome back from the break. We are going to look again at the discovery of some major human pathogens. We are going to look at the year of discovery. We are going to look at the particular disease that were discovered during that year, the causative agent, and the discoverer. In 1876, anthrax was discovered by Sir Robert Koch, and the causative agent is Bacillus anthracis. In 1879, gonorrhea was discovered by NASA, and the causative agent is Neisseria gonorrhoeae. In 1880, typhoid fever was discovered by Gafke, and the causative agent is Salmonella typhi. In 1880, malaria was discovered, and the discoverer was Leveran. The causative agent is Plasmodium species, with Plasmodium ovale, Plasmodium malariae, and Plasmodium vivax. In 1882, tuberculosis was discovered by Sir Robert Koch, and the causative agent is Mycobacterium tuberculosis. In 1883, cholera was discovered by Sir Robert Koch, and the causative agent is Vibrio cholerae. In 1883 to 1884, the diphtheria was discovered, and the discoverer was Klebs and Loifer, and the causative agent is Corynebacterium diphtheriae. In 1885, tetanus was discovered, and the discoverer is Nicoya and Kitasato, and the causative agent is Clostridium tetani. In 1886, pneumonia was discovered, bacterial pneumonia, and the discoverer is Frankel, and the causative agent is Streptococcus pneumoniae. In 1892, gas gangrene was discovered, and the discoverer is Welch and Nuttall, and the causative agent is Clostridium perfringens. In 1894, plague was discovered, and the discoverer is Kitasato and Yersin, and of course the causative agent is Clostridium is Yersinia pestis. In 1896, botulism was discovered. It was discovered by Van Imingen, and the causative agent is Clostridium botulinum. In 1898, dysentery was discovered by Dr. Shiga, and it is caused by Shigella dysenteriae. We have different species of Shigella. We have Shigella dysenteriae, Shigella sonnei, and Shigella boydii. In 1901, yellow fever was discovered by Reedy, and it is caused by flavivirus. In 1905, syphilis was discovered by Scaudam and Hoffman, and the causative agent is Treponema pallidum. In 1906, whooping cough was discovered, and the discovery was Bordet and Gongu, and the causative agent is Bordetella pertussis. In 1909, Rock Mountain spotted fever was discovered, and the discoverer is rickettsy, and the type of infection and the causative agent is Rickettsia rickettsii. Let us look at the summary. We looked at Anton van Leeuwenhoek as one of the discoverers of the who are critical in the early development of microbiology. With Anton van Leeuwenhoek, we have Sir Robert Koch, we have Edward Jenner, we have Louis Pasteur, we have Sir Alexandra Flemming and Joseph Lister are among the people who made greater contributions to the field of microbiology. Until tomorrow, thank you so much. Until we meet next time.